I'm very excited to now introduce Dr. Eve Richardson. Eve will be presenting the B-cell epitope prediction tools. Thank you so much, Eve.
Thanks, Nina. Let me just share my slides. Okay, so yeah, as Nina said, I have the privilege of talking to you today about our B-cell epitope prediction tools. So this is an outline of what we'll talk about today. We'll first have a very brief overview of B-cell epitope biology and sort of some insights into the possible features we can use for prediction. And then I'll talk about the prediction tools that we have available on the IEDB. And then in the third and final sections, we'll talk about the two types of B-cell epitope prediction methods that you can do on the IEDB. And I'll walk you through some examples. So the purpose of this is to familiarize you with the outputs that you might expect to see when you use our tools. So B-cell epitopes are epitopes that are recognized by B-cell receptors or antibodies. So B-cell receptors are the membrane-bound form of antibodies, which are these immune proteins. And these are heterodimers, and there'll be probably a familiar Y-shaped protein to you that you can see here with a heavy chain and a light chain. And the diagram is also showing that this is a modular structure where you have variable regions on sort of the arms of the antibody, and then you have the constant regions. And the variable regions of the antibody are the business end when it with regards to binding to antigens. So it's the variable domains that engage the antigen, and the majority of the contacts that they make with antigens are within the hypervariable loops at the ends of these arms, and those are called complementarity determining regions. So the epitope is the part of the antigen or the subset of the antigen that is recognized by the antibody. And you can have multiple epitopes on an antigen recognized by different antibodies. So this is an example of, so you can visually see how antibodies recognize an antigen. So this is an example of a complex that was solved by X-ray crystallography. And we have an antigen. In this case, this is hemagglutinin, and you can see the antibody in blue and pink there that's binding via the variable domains to the HA1 chain of hemagglutinin. And so the subset of this antigen that is in contact with the antibody is referred to as the epitope. So that's colored red for you there to see. And then on the other side, you have the paratope. And looking at this, you can probably immediately see that there are differences with regards to how this works versus how T cells recognize epitopes, and therefore what regions of the antigen are likely to be antigenic. So you can see that the antibody is binding to residues on the surface of this protein, for example, whereas for T cells, that can be anywhere within the protein. And so there are certain features that we know from looking at examples like this of X-ray crystal structures that there are certain features that are associated with recognition by antibodies, and probably the biggest one is surface accessibility. So that's really key for prediction here. So distinction that we often make is between linear and so-called discontinuous epitopes. So if you look at crystal structures of antibodies and antigens, and you actually look at the epitope residues and how they're sort of distributed within the sequence, you can see that there's two main classes. So with the discontinuous epitopes, you can see, so again, the epitope residues are colored in red, that you can have residues recognized that are physically close in space due to the fold of the protein, but they're non-contiguous in sequence. So they may be separated by many residues in sequence space. So these are called discontinuous epitopes or sequential or conformational epitopes. Conformational because they depend on the actual conformation of the protein. You also have linear epitopes, and these are epitopes where the residues are sequential in sequence. And more than 90% of B-cell epitopes are estimated to be discontinuous. And so obviously this fundamentally changes the way that we do prediction for B-cell epitopes versus for T-cell epitopes. So now with that, hopefully slightly useful primer as to B-cell epitope biology, we'll move back to the IEDB and look at the prediction tools that we have available. So if you go onto the IEDB analysis resource and click onto B-cell tools, you'll see this page, and we're going to talk about a subset of the methods that are available here. So prediction of linear epitopes, and then our two methods that are currently available for prediction of discontinuous epitopes using protein structures. But first, a very fundamental question is, when might you do B-cell epitope prediction? When should you do it? So I'd say data is always better than prediction. Prediction is always trying to get you to the point of data. So the first thing that you should do is verify that whether there is information for your antigen in the IEDB. So if people have previously reported epitopes for your antigen, that's the gold standard of information that you can have. But say that there aren't any epitopes available in the IEDB, nobody's ever looked at epitopes on your antigen of interest, then epitope prediction methods are what you should be going for. The second thing is there may be epitopes available for you in the IEDB, but you want to know all of the candidate epitopes that could be possibly recognized on your antigen of interest other than those in the IEDB. And again, in this case, you should look to prediction methods. So we make a big distinction in B-cell epitope prediction as to the input features that you will use. So there's sequence-based epitope prediction, and there's also structure-based epitope prediction. So for sequence-based epitope prediction, you don't input a structure or a structural model of the antigen. You just have the sequence, and a lot of the predictive power of methods comes from being able to predict which of those residues are surface accessible. So sequence-based epitope prediction methods have been used for many years, but recently things have changed somewhat, obviously, with methods like AlphaFold, which mean that it's very likely you'll be able to find a good structural model for your antigen. And then also as more and more 3D solved structures become available, increasingly through methods like cryo-EM. In that case, you will already be able to know with some degree of accuracy which of your residues are surface accessible, what your secondary structure looks like in each of those regions, what residues are physically close to one another. And in that case, we would recommend using structure-based epitope prediction methods. So now I'm going to talk more about these sequence-based epitope prediction methods. So the first class of methods that we have available on the IEDB are the amino acid scale methods. So basically they're a set of amino acid scales, which is essentially some value attributed to a particular type of amino acid. And then going over windows of the sequence, so consecutive chunks of residues of usually 5 or 7 residues, you have some method of, is mostly averaging over the window, but one of the methods uses some form of product so that you get, for a particular window centered on a particular residue, a value that correlates with one of these features that we know to be correlated with antigenicity. So, for example, the propensity for amino acids to form beta turns. Hydrophilicity is a really big one. So I didn't touch on this previously, but there are known amino acid biases within epitopes due to the fact that B-cell receptors themselves are very limited in sequence space. So you do see an enrichment of hydrophilic residues among epitopes. There's also measures like flexibility. And then, of course, surface accessibility is also a really important feature, as we've already discussed. And then finally, we have antigenic propensity, which is some measure of the frequency of residues in epitopes. versus non-epitope residues. So the sequence scale methods are these simple methods of combining things we know about amino acids across windows along the sequence. We do also have more complex methods, machine learning methods available on the IEDB, and these are the tools that we would recommend that you use. So for the machine learning methods, there's curated positive and negative training data sets. So positive being epitopes and negatives being residues that have not been observed in epitopes. And then you use features like the amino acid scales, so like the surface accessibility or the hydrophilicity. You use those as input to more complex algorithms that can learn from the training data that you provide to it, more complex patterns that are can produce more accurate predictions. So the machine learning algorithm for sequence-based epitope prediction that we currently recommend on the IEDB is BepiPred 2.0, and that uses a random forest. But if you're familiar with the literature, there's quite a large variety of different machine learning algorithms that people have used for this task. But yeah, in summary, these machine learning models allow you to learn more complex patterns that can distinguish your epitope versus non-epitope residues. So now I'll talk you through what it would look like if you went to go and use our linear epitope prediction tool within the IEDB. So if you return here, and then this is the relevant section. So the input, you can either input a Swiss prot ID or you can input your protein sequence in plain format. And then you'll have a choice among the prediction methods. So yes, BepiPred is the default and that is the recommended. Though, as we will see, it can be interesting to look at the other prediction methods to try and interpret the more complex machine learning predictions. And if you would like to learn more about each of these methods, there's links to the help tab. So this is what you'll see if you click through and look at the help and the reference tabs. And this is really comprehensive information about how these things are specifically implemented in the IEDB. So some condensed information from their papers, but we also provide the links to the papers if you have specific questions that are not answered on our help and reference tabs. So say, for example, we input the RBD region from SARS-CoV-2 spike glycoprotein, or we can input the sequence, or we can use a Swiss prot ID like here. We select the BepiPred linear epitope prediction 2.0 and click submit. And the calculation on this is really quick. The top of the page, you'll see something like this, which is the graphical representation of the predictions. So for each of these points, this is the prediction for the window centered on that residue. There's also this adjustable threshold. So by default, it's set here at 0.5. And then there's a coloring to help you interpret it, that where green, this is below the threshold. So these are not regions that you would predict to be in the epitope, and then yellow are regions that you do predict to be in the epitope according to your threshold. But you can adjust that yourself to increase or decrease stringency, depending on the number of predictions you'd like to make. And then if you scroll down, you'll see this table of the predicted peptides. And so those are the contiguous sequence segments where the score is above the threshold. So each of these black boxes corresponds to one of these peptides. You can see here that this is the longest predicted peptide of length 38 in this region. The other way that you can look at the predictions is the residue scores themselves, which you can download as a CSV file. So this is the tabular format, and you can sort by score, sort by assignment and position, etc. And the greatest flexibility you'll have will naturally be by downloading this and manipulating it in your programming language of choice. As I said, while we do recommend BepiPred 2.0 as a prediction method with the highest accuracy in unseen data, it can be productive to look at prediction results according to different methods in order to interpret that and identify the most consistent regions across different methods. So, for example, we see a spike here that we also see in BepiPred that we see in this beta turn prediction and that we also see in the flexibility prediction results. So that can help interpret your results. So having looked at the linear B-cell epitope prediction, we'll now look at the two methods that we have available for B-cell epitope prediction using protein structures. So this is a graphical overview of the two methods that we have. So we have the DiscoTope family of methods, and we have Ellipro. And I'll talk more about each of these in turn. But briefly, DiscoTope is this neighborhood-based method where for each of the residues you look at the neighborhood of those residues and aggregate amino acid log odds scores that you've calculated from some external training data, as well as the surface accessibility according to the number of contacts in the neighborhood. And then Ellipro is an orthogonal method that for each of those residues calculate some measure of protrusion, and then has a couple of other parameters that you can play with to cluster those actually into discontinuous epitopes. So DiscoTope was trained on 75 crystal structures of antibody protein complexes. The first iteration of this algorithm was in 2006, and it was updated in 2012. So DiscoTope 2, they both have broadly similar algorithms where each residue is assigned a value based on a linear combination of normalized values, so log odds ratios, and then surface accessibility. And I should say that this does represent a significant performance improvement over comparable sequence-based epitope prediction methods. So we're talking about around a 0.6 AUC for the sequence-based methods to around a 0.7 ROC AUC reported for DiscoTope 2. So this is a significant improvement. So let's go back to the IEDB and see how that looks. If you click through onto DiscoTope, you can see it's structure-based. And the fact that it's structure-based means that you need an input structure. So you want to identify either the ID of the corresponding entry in the PDB, or you can upload a PDB file of your own. You currently can only make predictions on a single chain at a time. And our recommendation for circumventing that is to actually manually, if you have like a multimeric protein that you want to make predictions on, if you just change the chain ID in your PDB file, I think it's column 22, to have one chain ID and run predictions on that. That's our recommendation for the multimeric proteins. But of course, you have to identify your a PDB structure to input. So we'll just briefly talk about how you can use the PDB. So this is an overview of the site currently. So there are over 200,000 structures, but you'll see that there are many more computed structure models. So those are models that have been produced with AlphaFold. And you can decide whether or not you want to use those models using this toggle include computed structure model. So we recommend using the sequence similarity search, so which will lead you through to this advanced search query builder, where you can input your sequence, and you can also toggle for whether you want to include your computed model structures. And this is a potential output for two different searches. So you'll see, according to whether you have this beaker or if you have a screen, whether this is an experimentally solved structure or whether this is a computed structural model. And the gold standard would be to use an experimentally solved structure, but for many proteins, unfortunately, there are no solved structures, and in that case, you should resort to using the AlphaFold 2 CSM, provided that this looks to be of sufficient quality. So when you search in the PDB, you can say we found our protein that had been solved at PDB ID 1LVH. We can download that PDB file, and then we also want to make sure that we are providing the correct chain ID. So you can click through in the annotations in the PDB and see in this case we could input chain A or chain E. It's slightly more complicated currently for the computed structure models. So if you actually go onto the PDB and identify a computed structure model as here, currently you cannot download a file format that would be accepted by the IEDB. We have the currently you can only download model CIF files. And yes, CIF files are not currently accepted by our tools in the IEDB. You can use it to check the chain, and this is A in monomeric proteins. To actually retrieve a PDB file that you can use, you can click through to the AlphaFold DB link, and there you can download your PDB file. So you have to make slightly more go through slightly more websites, but you can still easily retrieve a file accepted by the IEDB for this, so you can download your PDB file. So yeah, this is an example if we were to input by PDB ID 1Z40, and then we want to select our DiscoTope version. So we would suggest selecting DiscoTope 2.0. And you can, as with the other tools, you can modulate your scores according to the sensitivity and specificity. So increasing stringency as you increase the threshold score. So this is what the example output would look like for 1Z40 chain ID A. So it's a similar visualization as we had for the linear epitope prediction, where each of these points is the prediction centered on that residue. And again, we have the threshold, which you can change here, and the coloring of the residues according to that threshold. And again, the option to either do a table view or download that as a CSV. So this is an example of what the table view would look like for this prediction. And so you have the DiscoTope score here that you can sort by, and then there are also interesting other columns that you can use to, again, interpret the more complex scoring function. So you can look at like the contact number, which is a measure of how buried a particular residue is, or you can look at the amino acid propensity score, for example. And yes, the positive predictions are displayed in green. And then there's a nice add-on here, which is that you can look at this antigen in the 3D viewer. So this is what this would look like in the 3D viewer, and you can see that yellow here is the predicted epitope on the surface, obviously. And so that can be useful for quickly visualizing predictions or for making figures. And so after having looked at DiscoTope, we'll now just quickly talk about Ellipro for the final part of the presentation. So Ellipro is our other structure-based B-cell epitope prediction method. So it can predict both linear and discontinuous epitopes, and it's all based on the geometrical properties of the protein structure. So it's a combination of three sub-algorithms. So you approximate the protein shape as an ellipsoid, and then using that ellipsoid representation, you calculate a protrusion index for each residue. And then you apply some cutoff, and then you can cluster the neighboring residues. And so there are a couple of parameters that you can change to produce different sizes of epitopes, for example. So this is what it looks like on the IEDB analysis resource. You again can upload your PDB file or enter your PDB ID. And then there are two parameters here that you can change. So you can change the minimum score. This is the minimum average, sorry, the minimum score for a residue to be considered to be an epitope. And then you can change the maximum distance. So as you, this is part of the clustering algorithm within Ellipro. So that specifies a maximum distance between between epitope residues that are clustered into the same discontinuous epitope. So that's by default 6 angstrom. You can increase that to sort of produce larger discontinuous epitopes, or decrease it to produce smaller discontinuous epitopes. Yeah, so as above, as previously for multimeric complexes, those calculations will be made for each monomer independently. And so we recommend that you give each monomer the same chain ID if you want the prediction to to incorporate information about all of the chains in the file. So if we select the PDB chain here that we want to use, say the A chain, we get this kind of output. So where you have a table of predicted linear epitopes, and these are the consecutive regions in the sequence that are predicted to be epitopes, and then you have a separate section which is a prediction of the discontinuous epitopes, and those are provided separately. Again, you can view those within the 3D viewer to see where they sit in the 3D structure, and you can produce a similar like 2D score chart as we had for the previous methods, and that can provide some further information. So I'm at the point of summarizing now. Hopefully I've conveyed to you that we have numerous B-cell epitope prediction tools that rely on different methods from within the literature. Our recommended sequence-based epitope prediction tool is BepIPRED 2.0. Obviously, as times are changing and we have more structures and more good models for antigens, we would recommend to use structure-based methods, because that means you don't have to guess about which residues are close to one another and which ones are surface accessible. And we have two of those methods available, DiscoTope and Ellipro. And finally, the PDB is a really useful resource for finding your structures to input, as well as finding computational models if your structure unfortunately hasn't been solved yet. That's everything from me, and I look forward to answering your questions. Thank you.